Hi everyone, welcome to chapter 11, part 2. So in this chapter, you'll remember that we are discussing cell interactions. But one quick note before we move on, in our first lecture, if you're watching this in the fall of 2020, I misspoke. You do have a homework that is due this Friday, but that will actually be October the 2nd, not the 3rd, okay? And if you check that out, remember that it's due that morning by 8 a.m., okay? After that time, I start deducting points. You can still turn it in, and you should still turn it in for partial credit, but remember that I do deduct points after that time. Okay, so when I left you at the end of chapter 11, part 1, we had started talking about how cells communicate with one another. And we left off talking about how next door neighbor cells communicate. So for example, we had here an example of an animal cell. And right here we have a plant cell. And I told you that much adjacent next door neighbor type cellular communication occurs via the action of these tunnels. And they just have different names and a little bit different structure if you're talking about animals or plants. So you'll recall that we have what's called gap junctions in animals. And gap junctions are composed of these proteins that I've got drawn for you here that form tunnels by inserting themselves into the plasma membrane of each cell and then linking up, okay? So this can do things like allow ions to pass through or other molecules to pass through that can act as signals from one cell to the other. In plants, there's also a tunnel system. It's a little bit different in its structure. So you can see here that it's literally a tunnel. There really is not, here we are, it literally is not any kind of protein insertion. It's a legit tunnel through the cell wall. And it is actually often got a little bit of smooth endoplasmic reticulum threaded through it. The term for this particular structure is a plasmodesma. And remember that I told you that if it's plural, the term is plasmodesmata. Okay, so now let's move on to discuss how distant cells communicate. So in thinking about cellular communication in general, I want us to think about the results of cellular communication, whether we're talking about adjacent next door neighbor type cells or distant cells. We can really break down the results of cellular communication into two different results, okay? The first result is that signals from cell to cell, so I'm just going to put signals, they can cause changes in something called gene expression. Now, what do I mean by gene expression? Well, you know that in your nuclei, you have a whole bunch of genes, and those genes function as an instruction book. That instruction book is literally the instructions on how to make an entire organism. But let's say that you are a liver cell. You don't need to access the part of the instruction book that tells you how to be a brain cell. You only need the set of instructions relevant to your job as a liver cell. So there are some cells that are some genes that are turned off and some genes that are turned on. Sometimes the genes that are turned on or off can change with your situation. So for example, if you are experiencing a fight or flight reaction because you're being chased by a bear, there might be some genes that are turned on that allow you to increase your heart rate, and they sort of move your blood flow away from your digestive tract toward your muscles. And that's the result of turning some genes on and turning other genes off. So we call that process gene expression, but you can think of it as turning genes on or turning them off. And sometimes that happens as a result of signals being sent from one cell to another. All right. Another thing that cellular signals can do is they can activate or they can inactivate proteins, okay? Proteins that are already present in the cell. Maybe they're turning an enzyme, maybe the signal is turning an enzyme on or off, okay? So one thing that we're going to focus on as a mechanism, let me change this. One thing that we are going to focus on as a mechanism for activating some proteins is that we can add a phosphate group through a process called phosphorylation. And sometimes the addition of a phosphate group causes a conformational change in a protein that turns it on, okay? And so sometimes this process here will happen as a result of a cellular signal. The opposite can happen as well. A cellular signal can turn proteins off. So in general, there are two types of results that you can get from cellular signals. You can either turn genes on or off, or you can turn proteins on or off. So now let's think about how distant cells communicate. Okay, and in thinking about how distant cells communicate, it's got everything to do with cellular receptors. So first of all, distant cells are going to need to communicate using some molecules, right? And in multicellular organisms, these molecules are going to have to be distributed through some mechanism, whether it's a circulatory system, if you're like a vertebrate animal or an invertebrate animal, or if you're a plant, maybe there's a network of xylem and phloem tubes, which act kind of like a circulatory system in plants. But in any case, there are two basic types of molecules that often function as cellular signals, and you've heard of both of these. Hormones. So you might have a hormone produced in one organ that gets sent to another part of the body to give a signal. And the other type is a neurotransmitter. Neurotransmitters are better focus on my spelling here. They are molecules that communicate among brain cells in your brain to turn your neurons on or off, okay? We're gonna focus today primarily on hormones as a mechanism of distant cellular communication, all right? Now, typically what happens to hormones is that they bind to receptors. So let's review what a cellular receptor is. And we've mentioned cellular receptors before, okay? So I've got you some notes down here. Let's take a look at them. Oops, come on, little notes. I know we'll make that easier. Let's make that easier on me that way. Okay, so receptors are proteins on a cell surface or interior that receive a signal. So let's kind of review and go back to a slide that we've seen previously. This is a slide from chapter 7, okay? This slide, if you remember, covers the nuclear localization signal, okay? And just in case you don't remember, the nuclear localization signal was a series of amino acids that were tacked onto a protein that acted like a cellular zip code, telling the cell that this protein is destined for the nucleus. But what happens is these amino acid zip codes fit into another protein perfectly, okay? So that is a receptor. Now, This receptor fits into this receptor, and we form what's called a complex. And then this complex fits into the nuclear pore. So we've had this concept of receptors before. I kind of like to think of receptors as Legos that fit together. So you have a protein here that has the perfect Lego conformation to fit into another protein. And that's kind of a good way to think of hormones. So hormones might have a particular shape. And because they have a particular shape, they can bind to other proteins. And it's these proteins that fit the hormone shape that are called cellular receptors. And as I mentioned, these receptors can either be inside the cell or on the cell's surface, like on the cell membrane. Okay? When the hormone binds to a receptor, what typically happens is that it causes a conformational change in the receptor. Oops, holy smokes, don't know what happened there. Conformational change. In the receptor molecules, okay? And it's this conformational change that tells the cell to do something. And I'm going to show you examples of this, okay? So we're just kind of laying out a roadmap for how this whole process works. Now, the last thing that I want you to know is that different cells have different receptors, okay? Oops. Now, this is a really important concept. You know, if you release estrogen into your body because you're a female and you are producing estrogen, not every cell in your body has an estrogen receptor. Only certain cells have estrogen receptors, okay? So different cells have different receptors, and oftentimes that's what dictates how the cells function, okay? Let's look at some specific examples, okay? So the first thing I want to do is I want to distinguish between two different types of hormones that we're going to talk about today. We're going to talk about lipid-soluble hormones and we're going to contrast that with lipid-insoluble hormones. And this is how your book talks about them. But let's think about what the terms lipid soluble and lipid insoluble really mean. If you are lipid soluble, then that means that you are water insoluble, right? If you are lipid insoluble, that means that you're water soluble. So I just want to point that out because that's going to be important in a second. And the reason that we talk about this is because it has implications for how these hormones can get across the cell membrane or not. If you are lipid soluble, then that means that it's easy to cross a cell membrane, easy to cross membrane. That means it's easy to cross a cell membrane because cell membranes are primarily lipid. Conversely, if you are water soluble, it's going to be much harder to cross the membrane, right? If you're lipid insoluble but water soluble, it's going to be harder to cross that membrane. And just in case you don't remember, that was a chapter 6 conversation we had. When you examine the lipid bilayer and you look at what has an easy versus a hard time getting across, y'all remember that the interior of the cell membrane is primarily hydrophobic, okay? So it's lipids in here. And so like dissolves like. Lipid-soluble hormones can make it across this membrane, whereas things that are lipid-insoluble, hormones are usually pretty large molecules, they can't do it, okay? So let's look at an example of each type, and we're gonna start out with lipid-insoluble hormones. Let me go ahead and I'm going to insert a new slide here just so that you guys can have some room here for me to draw. And let me get rid of all this stuff. All right, so what we're going to look at for an example of a lipid soluble, I hope I'm not confusing you guys there, a lipid soluble hormone example is we're going to look at the action of steroid hormones. Now, y'all have heard of steroids before, right? What are they? Well, they are hormones. They're made from cholesterol, so they are lipids, and they are all hormones you've heard of. So for example, estrogen, and I'm just going to abbreviate estrogen, testosterone, and then you have a stress hormone called cortisol, okay? And there's another one that's a osmoregulatory hormone called aldosterone. All of these are steroids, okay? So steroids are all lipid soluble. They themselves are all lipids because they are derived from cholesterol. So how do they work? All right, let's draw ourselves a hypothetical cell. And today I'm going to use 2 lines to indicate the cell membrane so that we have a lipid bilayer, okay? So I'm actually going to draw 1 line, 2 lines, and just know that each line represents one part of the bilayer. So this is a single cell membrane, but I'm just not going to draw in all the phospholipids in the interest of time. Now, in this hypothetical cell, I want this side to represent the cytoplasm. So I'm just going to abbreviate that CYT. And I want this side to represent the outside of the cell. So in our example, let's just make this a human example, and we'll say that this is our bloodstream, okay? All right. Now, cortisol is a stress hormone, and it gets released when you are under stress. So let's just pretend that you are studying for an exam and you're very, very nervous about it. So your body starts to release cortisol, and it gets distributed in your bloodstream throughout your body so that it can communicate with distant cells. It's actually manufactured in your adrenal glands, which are right on top of your kidney, okay? But you want this signal to travel all throughout your body so that it can help you to do things like stay awake. Typically what cortisol does is it turns off systems that you don't need. So if you're under stress, you typically don't want to spend a lot of energy on digesting things, or you don't want to spend a lot of energy on immune processes. Instead, what cortisol does is it shunts bloodstream oxygen nutrients towards your muscles, because typically if you're under stress, you might need to get out of a stressful situation. So your body's still programmed to fight or flight, okay? So cortisol gets released from your adrenal glands, which are on top of your kidney. So I'm going to draw a little cortisol molecule here, okay? And I'm gonna label that C. Now the problem is your blood is water-based. Cortisol is a lipid. So it's not going to travel really well in your bloodstream by itself. So it's actually going to travel associated with a protein called a carrier protein, okay? But once this carrier protein arrives at a cell, it will release the cortisol, and cortisol very happily then crosses the cell membrane, okay? But then once we get inside the cytoplasm, we're in an aquatic environment again. So it turns out that there are proteins inside the cell that are there to receive this cortisol molecule. So if they are proteins there to receive the cortisol molecule, they are receptors. So let's draw a receptor here, and I'm going to draw it in its state before cortisol binds, and I'm going to draw it it's going to kind of look like a tooth, okay? So this is the receptor before cortisol binds, but once cortisol binds to this receptor, it's going to induce a conformational change. We're going to open up this little toothy area here, and here's my cortisol. So what happened? Cortisol bound to the receptor and induced a conformational change. Now, what this receptor will do, we have this what's called a hormone receptor complex, and I'm going to draw now a second set of membranes that are meant to represent the nuclear envelope. And I probably should have drawn this a little differently, because remember that the nuclear envelope is double membraned. So I hope I'm not confusing you, because I drew 2 lines up here to represent 1 membrane and then 2 lines here to represent 2. But hopefully you guys remember that the nuclear envelope is in fact double membraned, okay? And remember that the nuclear envelope has some pores that are studded within it, okay, called the nuclear pore complex. And so that's what this area here is, okay? So this receptor has a nuclear localization signal on it, and it's able to travel through this nuclear pore complex. Once inside the nucleus, what it does is, I'm going to draw some DNA here. It is going, can y'all see this is DNA? This receptor is going to travel to the DNA. It is going to find target genes that need to be adjusted, and it is going to work to turn those genes. I'm trying to draw this so it looks like it's bound on top of this DNA. It's going to work to turn those genes on or off, okay? So let's walk through this one more time, but let's do it using a little bit better graphic. So here we are. We have our steroid hormone, and it is bound to a carrier protein, and we're going to use cortisol as our example of our steroid hormone. This is a lipid-soluble molecule, so when the steroid hormone arrives at the cell, it's able to pass through that cell membrane. Once inside the cell, it binds to a receptor, inducing a conformational change. Boom, boom, okay? Now, once we have this conformational change, this thing is going to travel through the nuclear pore complex, which incidentally, you guys know that that means it must have, this must have a nuclear localization signal on it, right? Get rid of this right here. So once this entire unit makes it into the nucleus, it will find target genes, and it will work to turn those genes on or off to adjust the function of the cell so that your body functions under stress, okay? So what we have here is an example of, remember, this is a lipid-soluble hormone, and it had the effect of changing gene expression. All right. Now let's look at an example of the second type of hormone I told you about. The second type of hormone that I told you about is going to be a lipid insoluble hormone, okay? And there are many, many examples of lipid insoluble hormones. But since we're talking about stress, let's talk about another stress hormone. You guys have probably all heard of epinephrine. Some people may know it as adrenaline. Those are synonyms, okay? Both of those terms refer to the exact same hormone. And so this is a hormone that is lipid insoluble, which means that it is water soluble. So let's look at how that might travel through your bloodstream, okay? So let's draw ourselves another cell membrane. I'm going to make this a little longer and leave yourself a gap, okay? Now, again, this is a cell membrane, so I am assuming my phospholipids thusly, okay? Once again, we're going to treat this as the cytoplasm, and we're going to treat this exterior part kind of as the bloodstream, okay? So we have a molecule of epinephrine, and it can travel through the bloodstream all by itself because remember, the blood is primarily water. Epinephrine is lipid insoluble, but that means that it's water soluble. So it's absolutely fine traveling through the blood cell all by itself, but it's a rather large molecule. And if it's water soluble, that means it's polar. So it's not able to get through the cell membrane. Unlike our steroid hormones, it cannot travel through the cell membrane. So it's actually going to attach to receptor molecules embedded in the cell membrane. So let's draw one. These receptor molecules are going to look something like this. They actually go back and forth through the cell membrane 7 times. That's 1, 2, 3, 4, 5, 7. Did I do this right? 1, 2, 3, 4, 5, 6, 7. Okay. So these particular receptors are what we call they're 7-fold transmembrane receptors. But you can see that what's going to happen is that this fits beautifully there. Now, when this binds, it's going to cause a conformational change, and it's actually going to create a little kick. This little leg here is going to kick up as part of the conformational change. Why is that important? Well, it turns out that this receptor is right next door to a protein called a G protein. All right, so let's write that down. G protein. And a G protein has 3 parts, and I'm going to draw them kind of like this. It's got a part here, it's kind of half embedded. It's got a second part here, and it's got a third part here. And the third part here is attached to a molecule called GDP, okay? So all of this here is a G protein. This molecule is GDP. Now, remember how we had ATP, adenosine triphosphate, and ADP, adenosine diphosphate? Well, GDP is guanine diphosphate, okay? So it's the same G that you find in RNA and DNA. And what happens when this receptor kicks, it's going to kind of jostle this G protein, and it's actually going to break this third part off. It's going to break off that third subunit, and it's going to change this GDP into GTP. So what's happened? We kicked, we've broken this part off, and we have created a situation where we have GDP exchanged for GTP. Why is that important? Well, Let me erase a little bit right here. I'm going to redraw my membrane. Oops, come back. All right, redrawing our membrane. Well, it turns out that this complex here with the GTP molecule actually fits nicely into an enzyme here. And this enzyme is called adenylyl yeah, I'm spelling this right, adenylyl cyclase. Basically, we're going to take the phosphate group off of that GTP molecule, and we're going to use it to turn this protein on, okay? That process of turning proteins on using a phosphate group, I told you guys before that that has a name, and it's called phosphorylation. And I've had to break it up because I ran out of room, but it's one word. So we've turned adenylyl cyclase on. Now it's actually going to perform an enzymatic reaction. It's going to take ATP from inside your cells, and it's going to convert it to a molecule called cyclic AMP. And it does this over and over and over and over and over again. So what we've done here is we've accomplished a couple things. First of all, let's name this system. This protein here, this receptor, is called a G protein coupled receptor, or a GPCR for short, okay? And it makes sense because this is a receptor that's right next door to a G protein, okay? What we've done is we have taken an extracellular signal, and so I'm going to write extracell, and we've turned it into an intracellular signal. So we've changed this signal from extracellular to intracellular. That is a process called signal transduction. Changing a signal from one type to another is called signal transduction. So that's the first thing that you need to know about this. Oops. The second thing that you need to know about this is that the hormone acts as what we call the first messenger. And cyclic AMP acts as a second messenger. Now, what do I mean by that? Well, what this cyclic AMP is going to do is it's actually going to go throughout the rest of the cell, and it's going to act to turn proteins on and off throughout the cell. So what we've done here is we have gone and taken this first messenger and converted it into a second messenger inside the cell. The third thing that we have accomplished here is we have amplified the signal. Now, what do I mean by that? Okay, we had one hormone molecule up here, right? And it bound to our protein, our GPCR, and that caused a conformational change that kicked the G protein over, and that activated this enzyme. But once this enzyme gets turned on, it starts pumping out cyclic AMP, and we get lots and lots of cyclic AMP molecules for just one hormone input molecule, okay? So we've got one hormone input, but we end up with multiple cyclic AMP outputs. So we have amplified the signal. So let's walk through this with a little cleaner diagram just so that you guys can see. Okay, so here we have our epinephrine molecule, just like I drew for you guys, all right? And when the epinephrine binds, what happens is it creates a conformational change. It's going to kick this G protein. If you look here, you can see that the G protein has 3 units. Once we kick it, one of the units breaks off, okay? And what they don't show you is that you go from having GDP attached to having GTP attached, all right? So when we have that situation where the GTP and part of the G protein attaches here, we activate the enzyme called adenylyl cyclase. Adenylyl cyclase, once it's turned on, is going to convert ATP to something called cyclic AMP, okay? Cyclic AMP is going to act as a second messenger molecule that can travel throughout the cell to turn proteins on. Okay, so I'm just going to put on, and I'm going to put proteins. And since we're talking about stress, what are some of the proteins that might get turned on as a result of this epinephrine binding? Well, you guys know that whenever you have a lot of epinephrine or adrenaline, your heart rate increases. So you're going to have proteins or enzymes involved in increasing your heart rate. You're going to find your blood vessels open up to deliver more oxygen to your muscles, and you're going to start breaking down glycogen, which is a storage molecule, into glucose that you can use for cellular respiration. Now, here's the deal. How on earth does cyclic AMP accomplish these processes? And that has to do with the process of phosphorylation. So let me go here. This is the same system that I just showed you. So it's just drawn a little bit differently. So here's our epinephrine. Here's our G protein coupled receptor. Okay, epinephrine binds, causes a kick. The G protein goes from GDP to GTP, it moves over here, it activates adenylyl cyclase. Adenylyl cyclase converts ATP to cyclic AMP. Now, how does cyclic AMP cause all those actions I just showed you? Well, it has to do with that process of phosphorylation. So what cyclic AMP does is it binds to another molecule called protein kinase A. Protein kinases they are molecules that add phosphates to other proteins. I'm just going to put add P to other, to other proteins. Okay, so cyclic AMP comes along, it activates this protein kinase. This protein kinase then adds phosphate groups to enzymes and proteins that are involved in your epinephrine fight or flight response. So by adding phosphate groups, we are turning on proteins, okay? So let's circle back around for just a second and go back to my slide up here. Oops, no, this one, where I talked about the fact that cells communicate in 2 basic ways. There's 2 results of cellular communication. You can have changes in gene expression, and we saw that. We saw that with our steroid hormones. Or you can have changes in the activity of proteins. And we saw that with our G protein coupled receptors. Now, I know that these are difficult concepts to internalize. So what I want to do next, I'm going to pause this and I'm going to bring up a video that I have for you guys to sort of, I had a, where was it? I think it's down here maybe. Here we go. So down here, I have an animation video for you guys that on how epinephrine works. And we're going to view that together here in just a second. But I also want to let you know that there is an animation in your book. So if you go to the study area of your textbook and you go to animations, there is an animation in your book that demonstrates this as well. But I'm going to pause this, and when I come back, we'll preview this video together.
Epinephrine is one of many hormones that is water-soluble, hydrophilic, and therefore unable to cross the hydrophobic plasma membranes of its target cells. Instead, it binds to receptor proteins located in the plasma membrane and does not enter the cell. When epinephrine binds to beta-adrenergic receptors on the liver cell, G proteins on the inner side of the cell membrane are activated. Each G protein is composed of 3 subunits, and the binding of epinephrine to its receptor protein causes one of the G protein subunits to dissociate from the other 2. The G protein subunit which dissociates from the others carries a GDP, which is replaced by GTP when the subunit is activated. The activated G protein subunit then diffuses within the plasma membrane until it encounters adenylyl cyclase, a membrane enzyme that is inactive until it interacts with the G protein subunit. When activated by the G protein subunit, adenylyl cyclase catalyzes the formation of CAMP from ATP. The CAMP formed at the inner surface of the membrane diffuses within the cytoplasm, where it binds to and activates protein kinase A, an enzyme that adds phosphate groups to specific cellular proteins. In liver cells, protein kinase A phosphorylates and thereby activates another enzyme called phosphorylase, which converts glycogen into glucose 6-phosphate. The glucose 6-phosphate is then converted to glucose. Through this multi-step mechanism, epinephrine causes the liver to secrete glucose into the blood during the fight or flight reaction.
Okay, so hopefully you can see, let me change back to our...
Epinephrine is one of many hormones that is...
Sorry, let me go back to our PowerPoint. Let's see here. All right. Back to our PowerPoint here. All right. So hopefully that was helpful to you. Let's see here. This is it. There we go. Hopefully that was helpful to you to kind of understand the process. But again, please check out your book. Your book has a very good animation for both the steroid, and we'll say that that is the lipid soluble action, and then it also has a good animation for the G protein coupled receptor as well, okay, which is the lipid insoluble, okay? Now, I wanted to mention a couple things. So this is just a picture of that same system from your book, and so we're not going to go over this a third time, but I do want to mention that I gave you an example of cyclic AMP as a second messenger, and we talked about G protein coupled receptors. But there are other receptor types that kind of work in a similar fashion. For example, there's a section in your book that talks about something, an enzymatic receptor called a tyrosine kinase. I am not holding you responsible for that information, but I mention it in case you see it during your studies. It's another example of a receptor for a hormone that is lipid insoluble, oops, lipid insoluble, or another way of saying that is water soluble, okay? And different types of receptors create different second messenger systems. So I just wanted to point out to you that there are other second messenger systems that you'll learn about in other classes, maybe if you take biochemistry or if you go on to graduate school, okay? But one thing to point out is that these second messengers only occur when you have a lipid insoluble or a water soluble system. And the reason for that is because remember that the first messenger cannot cross the cell membrane. So you have to use a second messenger on the interior of the cell to accomplish your activation. So a great question would be something about like whether or not steroids, which are the lipid soluble type that can cross cell membranes, do they have second messenger molecules? And the answer is no, because they themselves can diffuse directly into the cell. All right, the last thing that I wanted to talk about, and I'm just going to mention it as something that you might discuss later on if you go on into microbiology. We've been talking a lot about how cells communicate in multicellular organisms, but let's focus a little bit for just a moment on how unicellular organisms communicate. So unicellular organisms are going to use some of the same concepts. So they are going to secrete substances, okay? Often these are going to be water soluble or lipid insoluble substances. That means there's going to have to be receptors. On the surface of the cell. So a lot of this is the same, okay? But what a lot of bacteria or unicellular eukaryotes do is something called quorum sensing, where they can tell how many organisms there are in a colony or how many organisms there are nearby. And so what they do is they secrete a protein that the nearby cells can sense using receptors. And when that protein reaches a particular concentration, then the bacteria or the amoeba or something will perform an action. So for example, there is a type of bacteria that lives in symbiosis with a squid. And when that bacteria reaches a particular population level or a particular concentration, it will start to glow. It will start to fluoresce. So that's a very cool symbiotic relationship. Here you have an example of what looks like a slug. So this looks like a slug and it looks like it might be a multicellular organism. But what it really is a whole bunch of individual amoeba, okay? And they built up to a particular population level. They secreted a bunch of proteins that were picked up by receptors on nearby cells. And once those proteins reached a certain concentration level, it caused the amoebas to come together in an aggregate form and form a slug-like body that can crawl across surfaces to get to other places. So I just want to bring up this concept of quorum sensing as something that you may come across again in the future. I would ask you to read a little bit about it in your text. There's only like a page or two of it, okay? And I am going to hold you responsible for that information for the purpose of quizzes and exams. All right, I hope that all of this was informative, and if you have any questions, please don't hesitate to ask. Talk to you soon. Bye-bye.